everybody. It's nice to see you all. It's great to see lots of people coming along to see what we've been doing. Hopefully it reflects all the different people that might benefit from our resource. So the way this is going to work today, I'll speak for about 35 minutes or so, and then we'll have questions at the end. If I can introduce myself, my name's James Stevenson. I work in the UniProt protein function development team here at EMBL-EBI in the UK. And I'll be presenting today with the lead developer on ProtVar, Prabhat Tatu. So obviously, importantly, the ProtVar wouldn't be possible without funding, and our funding comes from the European Molecular Biology Laboratory in partnership with Open Targets, which is a public-private target validation consortium. So I'm going to talk about ProtVar, and of course, ProtVar is all to do with the interpretation of how genetic variation affects proteins. So we have to talk, at least for one slide, about the biology of proteins. Of course, as most of you on this call knows, proteins are really the workhorse of biology. They do most of their work in the cell. We have about 20,000 unique proteins in humans, and the function of each of those unique proteins is defined by the shape of the protein. As you can see, an example of a shape of the protein down at the bottom there. The shape and chemistry is dictated by the sequence of the protein. And therefore, if we have a genetic variation which perturbs the amino acid sequence, which we call a missense variation, that can in turn alter the shape and then how the protein works and may then lead to disease. So the challenge really is that the effect of that variation is very position and context dependent. So much so that the same variation in two different parts of the protein can have entirely different effects. So interpreting how a variant in one part of the protein might affect a protein and in turn human health requires a broad range of data to be brought together and coherently assimilated. And that's what ProtVar tries to do. So we're hosted at EMBL-EBI and maintained by UniProt developers. And what we're trying to do is to help users to collect all the information that they might need to be able to assess how likely each of those missense variants is to affect the function of a protein and ultimately human health. It's free for everybody to use in academia and industry, and we hope lots of different people will benefit from it. And the people on this, the breadth of people on this call suggests that hopefully will be the case. So really, without too much further ado and too much more introduction, I think we should look at the resource itself, and I'll try to demo how it works. So the address you can see on screen here, ebi.acdk/ProtVar is the way you'll access the tool. Okay, so I think we can get rid of the banner advertising the webinar as you're all here. And the first thing you can see here is that we've got a huge paste box. This is for pasting your variants into to get annotations. You can see on the right here, there's a variety of different formats and data types that you can use. And that's one of the strengths of ProtVar, that there's many ways that you can enter it and retrieve your variations. The first one is using genomic coordinates. And this is a VCF format. Well, as a user of these kind of tools myself in research, I know how annoying it can be that if you've used the wrong type of tab or too many spaces and things don't work. So we try to be quite forgiving in ProtVar in the user input. For example, instead of a tab, maybe we could have a / here, or maybe we could just have a space. Maybe we don't have an ID, so we'll get rid of that column. And these are all valid inputs for ProtVar. We're able to handle both DRCH38 and GRCH37 assemblies in ProtVar. And of course, occasionally users won't be absolutely sure which assembly they have. And therefore, ProtVar also has an auto-detect functionality. And if I just show you how that works, if we were to submit these 4 variants, you can see at the top of the screen there that we've determined that the assembly version is GSH38. And in this case, 100% of the variants match that. So we match the reference nucleotides against the 2 different assemblies to see which one scores the highest. So going back here, we can also, as well as VCF, genomic coordinates, we can also enter in a kind of, this is sort of the format from Nomad's user interface, in case you just want to grab some variants in there and have a look. HGVS format as well. And quite differently, we also allow users to submit protein positions rather than genomic coordinates. Again, you can see here on the example, the flexibility of ProtVar in that you can use single letter or 3-letter amino acid codes. Also, you can enter spaces and you can change the order and it will be interpreted by ProtVar. So the way ProtVar interprets this kind of input is that it will retrieve the codon sequence at the position and accession, uniprot accession that you have entered. And once it knows the codon sequence, it can then consider what the variant amino acid is and then calculate which of the 3 positions in the codon is required to be changed in order to affect the variant allele. Of course, occasionally that might be 2 different positions or 2 different variant nucleotides, in which case both of them will be shown in the results. And the final way you can access ProtVar is via a dbSNP ID. So you don't have to paste. Of course, if you have a very large list, you may have hundreds of thousands of variants, then you may want to upload a file instead. And you can do that by pressing the upload file. And then if I just upload a file here, the file name will be listed here. As you can see here, I called it 800,000. So this is 800,000 RS IDs that are protein coding. And even a large file like that, if we were to submit it, it will take a second perhaps, and then you can see the results here, paginated 25 or however many you want at a time. But you can see that here there's 33,000 pages of results that you can go through. We can also add mixed inputs. You can't add mixed genome assemblies because it's too difficult to interpret which variant is which assembly, but we can add mixed formats. For example, the top one here is a genomic coordinate, then we have a protein one, an RSID, and then a different way of entering genomic coordinates. If we add this, then it's all handled by ProtVar, and you can see it gives you a results table which is uniformed. So this results table going from genomic coordinates to protein positions and back, we cover just over 92% of human proteins with our mappings. There's still some that we're working on to try to eventually get that higher, but at the moment we have a high coverage of 92%. And you can see, hopefully from this results table, that there are some pink boxes, and the pink boxes represent the user data type, the type that users entered. So in this case, in the first line, the user has entered a genomic coordinate. In the second line, they've entered a protein position, an accession. And in the third line here, you can see they've entered an RSID. And actually hovering over any of the lines shows you exactly what the user has inputted. This is a direct copy of what the user inputted. So you can see in the first one, they've inputted VCF type format, and in this one, an RSID. So the results in ProtVar are roughly split between genomic and protein information mapping, and then finally annotations. If I explain the first part, genomic, we can quite intuitively see that we have a chromosome coordinate, an ID, if it's been added. This doesn't have to be a dbSNP ID or a well-known ID. You can enter your own IDs in order to keep track of your variants, and they'll be displayed here. We have our reference alternative allele here, which, as you can see from the protein input, has been calculated by ProtVar. And then we have the gene name, which links to Ensemble, and we can see the sequence of the codon, and in capital letters, the nucleotide has been changed. At the moment, ProtVar just handles single nucleotide variants. We'd like to increase that breadth in the future, but that's what it does at the moment. That doesn't mean that if you enter a non-SNV, you won't be able to run your results. You still will. It'll just, in that particular line that you've entered an insertion or deletion, let's say, it'll just give you a warning that this is not an SNV and it's not able to be processed, that particular line, by ProtVar. So the next line across, you see there's a CAD score. This is a third-party pathogenicity score based on the nucleotide itself. And then when we get into the protein section, we can see this first part is isoform. So ProtVar always tries to map to the UniProt canonical isoform, because that's the numbering system used by UniProt for the annotations. However, we also do map to all alternative isoforms that we can. In fact, we have all of the transcripts and isoforms in every position in the human proteome pre-mapped, which is why the retrieval is so quick when you saw the 800,000 RSIDs being retrieved very quickly. And you can see in this case, although the protein position is 783 here in most of the isoforms, you can see in one of the isoforms it's at a different position. Okay, so then we have a protein name and position, and we interpret what the amino acid change is and the consequence. This final column here shows another pathogenicity score. This is the EVE score. This is an amino acid level pathogenicity score, which is different from the nucleotide score. And EVE scores, again, is a third-party pathogenicity predictor. These are just based on evolution from sequence alignments. And the different colors and categories can be seen by clicking on the view legends here. You can see on the left, we have the EVE scores, then the CAD scores in the middle, and then these annotations. So while this is the mapping, and mapping is entirely essential, of course, to be able to retrieve annotations, the actual kind of meat of the tool, the annotations themselves, are at the end here in these three buttons. The first button is functional annotations. The second one is population observations. The final one is the structure. So I'll go through each of those and try to show what ProtVar can offer you as far as annotations. So this first button, these are the functional annotations. It's split into different sections. This section on the left, this box on the left, shows information about the specific residue that you're interested in. So in this case, it's position 493. And you can see here histine to an arginine change at 493. We calculate a conservation score just based on alignments for every position in the human proteome that we can deliver to you. And then these next three parts, these are all retrieved. These are all annotations retrieved from UniProt. And these are, as you can see, curated from literature. So here we can see that this particular change, histine to arginine change in position 493, has been involved in a mutagenesis experiment with a 3,000-fold reduction activity. You're able to follow these PubMed IDs to find the evidence for that. And then here, a 15,000-fold reduction activity, and follow these IDs as well. The final thing in this, the residue position annotations, is a fold X prediction. This is pre-calculation through our collaborators in Open Targets, and this shows the change in predicted stability of the protein when you go from the reference to the variant allele. And you can see there's the change there. These are done on AlphaFold models, which is why we also have the model confidence at that position as well. We have other predictions from our collaborators as well on the right here. So this box on the right doesn't show annotations specifically for the position, but of the region around that position. So for example, here on the right-hand side under predictions, if we click here, we can see pockets containing the variant. This is a predicted pocket, and it's displayed here because our position of 493 is one of the residues that makes up the pocket. So this variant position is predicted to be in a pocket, and we have some numbers on the confidence on the pocket itself. Also, we have protein-protein interface predictions. So again, this is from experimental evidence that two proteins are interacting, but we, through the collaborators in Open Targets, predict the actual residues involved in the interface. And in this case, our variant 493 is involved in that interface. And for both the pockets and the interfaces, we show them on structures as well, which I can show you later. So as far as the region, we didn't have very much information from UniProt for that particular region, but clicking on a different variant, you can see while there isn't much information on the residue position itself, in this case, we do have quite a lot of information about the region. We can see that this particular protein is a transmembrane protein, and the region where our variant is in the cytoplasmic part of the transmembrane protein. We can see that it's in the a functional domain, a protein kinase domain here with the range and links to the rule. And we can see that there has been mutagenesis experiments on the region, although not specifically on our variant position. If we go further down in the functional annotation section, we can see more general information about the protein. This is taken from UniProt. Perhaps we've assessed that the variant is likely to cause some kind of functional change, and then we want to consider how that functional change might affect the human. This kind of information is very useful. So again, it's not specifically for the variant position or the variant itself. This is more general information about the protein from UniProt. And you can see there's general text, but if you go further down, we have some specific information about this entry. So for example, the sequence was last modified a long time ago, 1994, but the entry itself was last updated the 3rd of May, so quite recently. So this is the functional information, and the functional information is constantly updated. And you can see much more specific information if you're interested in the catalytic activity or to do with information on the complexes or PTMs, etc. You're able to click through these various categories and find more information from the links itself. Interactions, for example, you can go through to intact and see much more information about the different proteins that our variant protein interacts with. So that's the functional annotations tab. The next button is the population observations. This is used to explore whether the variant that you've entered has been seen in human populations before, or whether variants that are similar have been seen in populations before. So on the left hand side here, this box shows other resources that have your variants. So for example, ClinGen, ProtVar, exec, nomad, and links out to those resources and IDs to those resources. If there is any explanation of the potential consequence, that's also given on the left here. So this is about the change that you have entered, specifically natural nucleotide change as well as the amino acid change. On the right here, we have co-located variants. Whereas you may generally think about co-location of variants being at the nucleotide level, because of course the mutation itself is at the nucleotide level. ProtVar is very much centered at the amino acid level. And therefore the co-located variants you can see in the box on the right here is the same amino acid changing, not necessarily the same nucleotide change. So for this first example of the asparagine to the aspartic acid, if you look very closely, we can see that this is position 435, whereas our variation is 436. So it's actually the first position in the codon, but it is the same amino acid. And of course, it causes a different change. If we look at the second one, it's the same nucleotide position, but it's a different nucleotide change, an A to a G rather than A to a C, which means it's an asparagine to a serine change. And if we click through here, we can again find all the identifiers for that co-located variant and also diseases that are associated with that co-located variant with the asparagine to serine change. Right at the bottom of the screen here in the same population observation section, we can see diseases that are associated with this variant. So you can see here there are 4 different diseases associated, and we have all of the literature links that you can go through to, and the ClinVar link as well. So the final tab, the final annotations to help you try to understand what your variant might be doing is the structure tab. So firstly, we show a table here with all of the experimental structures pulled from PDB for your variant position. And that's quite important because these are not all of the structures for the protein, but all of the structures for the protein which contain your variant position. And you can see by clicking on it, you can see where the variant is, and we're able to use this viewer. You can make it full screen if you'd like. You're able to do things like take a snapshot. This could be useful. You can download a snapshot straight from here. If you want to consider the residues that are surrounding your variant, clicking on it will show the interactions. And then if you'd like to get a different view, we can also make the structure spin, and while it's spinning, we're able to change things as well. So hopefully that could be useful. We also, though, have predicted structures. So we have the AlphaFold structures for each of the human proteins. And of course, there may not be experimental structures sometimes, so the AlphaFold models could be particularly useful there. You can see that the structure is colored by the model confidence from AlphaFold, and we also have the predicted align error from AlphaFold as well. And you can click through to the AlphaFold page for this particular protein to find more information. Sometimes when we have pockets, I mentioned the pockets before, this particular one doesn't have any, but if we go down here, we can see when there are pockets, then we're able to hover over and see the pocket location. Here it highlights all the residues that are in the pockets and your variant. Okay, the final parts of the structure section is the the protein-protein interfaces. And you can see here, sorry, I should go back to, here we go. So here, these are the models for the protein-protein interfaces where your variant is at an interface. So you can see in this case, you have your variant here, and if you hover over the residues that are for the interface, you can see in pink the highlight of the interface residues, and our variant is one of the interface residues here. So then you're able to have a look, and you can see it in context with the other interacting protein. Okay, so this information might be useful to you, but having it all just on screen on the UI might not be great for your future work. So of course, you might want all of this information downloaded. To do that, we go to download results in the top right-hand corner here. Here we have further options. We might just want the mappings only. Like I say, we've pre-computed the mappings. It's very quick to retrieve these. So we can just get all of the mappings from genomic to protein positions and back again. Or perhaps more likely, you want annotations. And again, we can just choose functional annotations. We can choose population or structure or all three. If you'd like to add an email, that's optional. But for a particularly big file, it might take a while, and we can email you when it's ready. And you can put a job name here. So if I just put a job name like a webinar test, we submit, and then going to the My Download section on the left-hand side, we can then see all of the jobs. Here's the webinar test that we just submitted, and from there we can download to our local machine. And My Downloads contains all of the things that you've requested for download during your session. So you can submit lots of different variants and different types, and they'll be all be stored here. So, of course, download is not the only way we can get information from ProtVar, and I'm going to hand over now to Prabhat, who can explain more about how we can access ProtVar programmatically. So, Prabhat, over to you.
Thank you, James. Let me share my screen. Okay, so yes, so in this part of the webinar, I will briefly cover other ways of accessing or linking to ProtVar. I will talk about the ProtVar REST API that provides a programmatic way to obtain information from ProtVar. The ProtVar website that James has presented is backed by the same REST API. So we are hoping users will find it useful to access data directly, for example, maybe to integrate in their analysis pipelines. On the website, we are showing one possible way of presenting data. There are different other ways the user may want to present the data to interpret information in specific ways. So the API is useful to get specific tailored information quickly, so you can embed it into your own pipeline application or algorithm. Let me go back to the ProtVar website. So from the ProtVar website here, there's a link to go to the REST API documentation page on the top navigation. Click on it and it will open the API documentation page in a new tab. So there is a list of endpoints in alphabetical order on this page. So each endpoint retrieve specific information. Depending on the user's experience, programmatic access can be made in a number of ways. For example, via command line using the curl command, or from your Python script or Java application. You can also use a graphical tool like Postman to send post requests. So I'm going to shortly show how to make requests directly from this page as well. The most important endpoint here is perhaps the mappings endpoint that does the mapping between genomic coordinates and protein position. So it is a single endpoint that allows the user to enter inputs in a number of different formats, as James has already gone through. This is supported at the API level. You can try it here directly on this page. So there are some input examples you could use. In this case, it is a mix of genomic input, the first one, protein accession and position, the second one, and the last one is an example of a DBSNP reference number or ID. So the request body is required in this case, as specified in red here. There are a number of options the user can specify, including whether they want to retrieve, in addition to the mappings, the different annotations for function, population, or structure. But by default, these are not retrieved. By default, we do a lazy genomic to protein mappings retrieval only. I'm going to now click on execute to run this example and see the response. And as you can see, when you do this step, the API documentation page gives you the corresponding curl command that you can use to execute the request from a command line. And the response itself, which is in this box here, is in JSON format. The structure of the response is defined in the schema section at the bottom. If you scroll down, you see there's a schema section, and you'll see in the schema the mapping response, for example. This is important when consuming a REST API response. So for the main mapping, the data table is pre-computed, as James has already mentioned. So that's why the retrieval is fast. And for other data like the annotations, these are initially loaded on demand, that is lazily, in the results page on the website. This is so that we have up-to-date information, but once the data is fetched and cached, subsequent retrieval should be fast. So the other difference here on this consuming this API is that the user has a full list of all the results at once, and whereas on the website the results are shown 25 inputs at a time, and we can see that each input is mapped to a corresponding output in the mapping here. And for each input, you either get a list of mappings, as here, or an empty list, and you can check there is an error attribute here to find out what may have gone wrong with any particular input if you don't have any mapping. And for genomic input, as the first example that we have used here, there may be multiple mappings in cases where we have multiple genes at a specific genomic location. This could be either in the same strand or in both forward and reverse strand. And for protein input as well, there may return multiple mappings, especially if we have not been able to filter it down to a reduced set of genomic coordinates as we infer the possible genomic inputs from the user's protein input. So moving on, this was the main mapping endpoint example. You can try out the other endpoints, the get requests in blue here, directly on the browser, for example, to retrieve amino acid level annotation. This one, you can use this links, for example, to see the raw JSON directly in the browser. And of course, you can refer again to the schema to understand how to interpret this this response in your client. So this example is for functional annotation information for this specific protein and at this position. And we have endpoints related to sending download requests in this section here. And if you have to submit a file input instead of text input, you can monitor the progress of your download using the download status endpoint. And there's also the first endpoint here is the assembly mapping or conversion endpoint, which is used to convert between different assembly builds. At the moment, we support G, R, C, H, 37, and 38. And we have a pre-calculated equivalency table for all protein coding regions. The last group of endpoints is for predictions, including retrieving conservation scrolls and pockets and protein interaction, as James has already gone through on the website. The next part I was going to quickly mention is the direct access, direct link. So you can access variant annotations directly using a permanent link URL by skipping the input screen on the website. This may be useful if you want to include reference from your own resource to ProtVar. In this way, by either using the genomic coordinates or protein accession and position, you can link to ProtVar directly. You can see in the first example here, on the top right, you can build a URL following the usual convention of specifying query parameters, in this case using the genomic coordinates attributes by specifying the chromosome, the genomic position, reference, and alternative allele. And if you copy this and open it in the browser, so this link is a permanent link to this variant, and it can be shared as well. The second example is using protein attributes, accession, protein position, and reference and variant amino acid. And this again, if we try it out, retrieve just the results for this protein input. So more details for this is available in the help section here, which I think James may go a bit more into details, but for the direct link, you can see how to use it here. Yeah, I think that's mostly my part. I will hand back over to James now, who will carry on and summarize the main points. Thank you.
Okay, hopefully you guys can see my screen again now. Yeah, from talking about the website and talking about the programmatic access, hopefully we've covered a lot of what ProtVar can do. But if you're still stuck with anything, or there's any particular parts that you want my help with, The ProtVar help section is quite big here. You can see, going to the help, we can look at the direct variant links API, which ProtVar already talked about. But then there's quite a lot of help on the website. We have these pages which shows you each part of the website and each field explained. And then if you want even further information about it, there's more detailed information here as well. And from there, there's also links out to the original sources. So the papers and the websites that we get the information from. Additionally, we have quite a comprehensive explanation of all of the columns in the download. So that when you download a file, there are 41 columns, which contains all the information that you saw in the UI, but having so many columns, it can be slightly confusing, and also the way we delimit lots of information in a single column. So here, step by step, we show each column and what you expect to find in it and how it's delimited. So that hopefully will be help to you. If you still need more assistance, or you want to make contact with us as far as suggesting things that you would like to see in ProtVar in the future, perhaps you can use the contact form, which I've clicked on here. Just add your name and email address and then submit to us, and then we'll get back to you as soon as we can about it. And then finally, if you want to be kept updated about the new developments in ProtVar, we hope to add new functionality on a kind of rolling basis, but we'll have major releases in line with the UniProt releases, which will be 5 or 6 times a year, where we'll refresh all of the data and all of the mappings, as well as adding new functionality. So if you want to know about what's new before anybody else, then you can add your email address here and you can subscribe. Okay, so yeah, that's it for the website. As a kind of summary of what we've shown you here, hopefully we've demonstrated here that ProtVar is quite fast because our pre-mapping's quite flexible with all the different ways we can access it. And we've got quite high coverage, like I say, over 92% of human proteins covered. We have a broad range of annotations as well as predictions to help you understand what varying an amino acid in your protein might do. We have co-located variants at the amino acid level rather than nucleotide level, with all the associated annotations with that. Interactive visualizations, which we're constantly trying to improve, of the experimental and also predicted structures and of complexes. Programmatic access, which ProtVar explained to you to help you reach the data more easily. And the point I made before is that it is maintained, it's updated, and we are actively developing ProtVar. So in the next release, which will be probably hopefully a few months' time, there will be new functionality to ProtVar, as well as expansions in the coverage and improvements in the visualization. So if there are things that you'd like in the next release, please use the contact page and say, it'd be great if you could add this, or I'm not sure how this works. Can you make it clearer? That's fantastic. As written at the bottom there, we want to offer everybody an intuitive and efficient resource so that you're able to interpret missense variation. So we really want it to help you all. So if you can, feedback now and then in the future through the contacts, we'd appreciate it. And then finally, any questions from the room? So if Daniel, you can handle that.
Okay, thank you very much, James and Prahat, for a very nice presentation and a demo. So yeah, we have several questions, so we'll try to answer, as we said, as many as we can. We'll try to go more or less in chronological order, but also for attendees, remember you can upvote by liking the questions if you have any in particular, and we will try to prioritize those. So the first question is, how does the engine recognize gene if only protein position is given? It could be for any gene. Thanks in advance.
So yeah, I mean, I may reply to this. So there may be input for which we have multiple genes at a specific genomic location, as I mentioned. So when we are providing protein input, there is a genomic coordinate determination step from the protein input. So just so this may be a range, so we may not be able to come to a specific genomic location. But even if we don't, for each of the genomic location that we come to, there may be multiple genes at that specific location. And we haven't shown a particular example in this presentation, but if you do enter a genomic location where there are multiple genes at that particular location, both whether it is in the same strand or in both forward and reverse strand, it is going to be displayed so we can see and interpret it.
Okay, thank you. Next question, can you add more than one variant per protein?
Yeah, absolutely. There's no limit on that. So as many variants in as many proteins. I mean, you could just enter 200 variants in the same protein, and it will display line by line. We keep the results in the same order as your input. So if, for example, we can't process one of them because we can't map it, it will stay in that position in the results. It'll just give you a warning. Equally, for example, if you add a reference allele which we don't recognize in that build, for example, we'll again give a warning and say, are you sure that this is the reference allele that you want? Because this is not the one that we recognize in the most recent build. But there's no limit. There's no limit to the numbers that you can upload, either via the paste or the upload file. Upload file is, I guess, only limited by the time it takes to upload on your server. But there's no limitation there.
Okay. And also, can you have the superposition of the protein structures of the original and missense mutation?
Yeah, that sounds. We'd love to do that. That would be a great idea. Of course, this was calculated locally in order to make the fold-X structure stability change calculations, but there's so many of them that we didn't keep all of the coordinates. I think it's a nice idea. This, of course, would be meaning keeping coordinates for 20,000 proteins, maybe 300 positions, and all 19 changes. It would be quite a big to do. But saying that, it's not impossible, and it's something we could look to do in the future if that's of interest to people. Yeah.
Okay, thank you. More questions. Can I make queries using R or Python package?
Robert, do you want to take that?
Oh yes, since this is a REST API, you can use any language, any REST client to query the API in whichever way you want. Yes.
Okay. Are the conservation scores the same as those calculated in VarSite?
Yes. Yes, they are. At the moment, we're using the VarSite conservation scores. So I was involved in the development of VarSite before doing VarMap. So we're using those conservation scores at the moment. But certainly in future versions, we can look at different conservation metrics. If there's any particular ones that you would like to add, then we can do that. At the moment, yes, they are the same exactly as they are in VarSite.
Okay. Is there an API that allows machine-readable submission and retrieval of information?
I think that is up to the client. I mean, this can be easily implemented if the user wishes to.
Okay. Are ETH scores available for all proteins?
So currently, we have ETH scores for about 3,000 proteins. The last publication from Deborah Marks' group, although the rest of them have been calculated. In fact, they've been recalculated for those 3,000 as well. So for most proteins, they should be available. We're just waiting for that publication, then we're able to have access to it. We've talked to her about that. So at the moment, only about 3,000, but very soon, in the next release, certainly we should have close to 20,000 proteins.
Okay. There was someone who said, I may have missed it, but how do we define functional domain?
So these are just annotations taken directly from UniProt. So the way UniProt would get information from Interpro, for example, they have PFAM domains and CAF domains. So functional, it like you say, it depends on the definition. These are either directly from UniProt, and we have a link where we have the functional domain. We have a link to the UniProt definition. So we have a rule, like a ProSite rule or the Interpro rule, and that will define how that particular domain has been called a functional domain. But we don't make any extra definitions on top of what's already been defined elsewhere.
Yeah. Okay. Yeah. Another question. For the direct link access, can I submit multiple position changes at one time?
No, I think for the direct link access, you can only submit one genomic coordinate or for one protein accession and position at a time.
Okay.
Again, if this is functionality that's useful specifically for you, then yeah, get in touch with us and we can look at how to do that. I mean, it get very complicated if we have very big ones, but if there's a group of 4 or 5 that you need to do, or even in the same protein, for example, that might be easier for us to implement.
So are bulk and specified millions, are bulk millions annotations supported in any way?
Yep. So I mean, we don't specifically have a limit. I mean, we have ourselves run hundreds of thousands or millions of variants. It depends. It's time really. So at the moment, the way that we retrieve data for the download means that if you were to submit 1 million or 5 million variants, it would run. It would just take longer. Maybe for millions of variants, 5 million variants, maybe it'd take half an hour to get the results back, that kind of thing. As far as on the UI, it wouldn't make any difference, really. You could submit 5 million and it would load immediately because it's lazy loading. So the functional annotations only appear once you click on it. So that wouldn't be a problem. You could get that back very quickly. But the download is where you would. So I think somebody made a comment about, for example, running Nomad and that kind of thing. So what we've done some of, and what we'd like to do in the future is we're going to make those kind of popular but very, very big sets available for download. So we're going to pre-compute, for example, the ProtVar sets and Nomad sets, so that you don't have to run it through ProtVar again, because we'll have it all pre-calculated for you just to download the file with the annotations on it. So I would like to handle the very big submission.
Someone said also, thanks for the nice presentation. Can you share what features data you are planning to add to ProtVar in the future?
Yeah, I mean, the next thing, there's lots of small things we want to make it faster. We're going to pre-compute all of the functional annotations, and so we don't have to call external APIs for that to make it fast. But as far as the new features, we want to do some more advanced filtering and searching, which means that if you want to, for example, pull all known variants which are in pockets, then you should be able to do that, or all variants that are in PTM sites or that have ligand contacts, those kind of more advanced searching techniques, rather than just pasting variants in. That's one direction we'd like to go in. But we have a list of 100 different things we'd like to do. It's about prioritizing. So especially, it's another good reason to contact us and suggest things that are really important to your work, and then we could prioritize those.
Definitely. Okay, thank you. Someone else who also said that the tool really looks amazing. And is it possible to download ProtVar and set up a local version, both front end as a viewer and back end?
Not yet. Not at the moment. Okay. It's something we've talked about, and it's something I would like to be able to do, especially when it comes to perhaps more sensitive variants that you don't want to submit over the website. So yes, it's something, but I don't know when we'll do that. In future releases, it'd be great to be able to have a standalone version as vectors, for example.
Okay. There's another question might be related. Is there a publication or link? Are pre-computed tracks downloadable without the API?
Oh, I'm not sure what it means without the API.
So at the moment, like I say, we'd like to have these big sets pre-computed and then retrievable. As far as our mappings table, that's another thing we potentially could make available if people found that useful, because we have the mapping table already pre-computed. So that could be downloadable. It isn't at the moment, but if it's useful to people, we could have that downloadable. In the future, when we do have them standalone, then of course all of the annotations will all come along with it, and they'll all be there for you as well. But certainly the first thing we're thinking of doing is trying to get these big sets, Nomad later when it has a bigger release, and places like ClinVar or other places that we're trying to get the big sets and make them available for download, so that you don't have to run the computation yourself.
I think it is also advisable, maybe if you want to download large data set, maybe try it without the annotation first, because this is very fast to do and to get. And then if you're trying it with annotation, maybe select the specific annotation that you want to in the output file as well.
Moving to more questions. Is there anyone where one can see if computational predictions have been confirmed experimentally?
Yeah, not at the moment. So like I say, for the protein-protein interface work, all of those protein-protein interactions have been confirmed experimentally. but yeah, it'd be good to put those links to those experimental verifications. As far as the pockets, no, there's no experimental verification for those, but they're both ongoing pieces of work with open targets. So as they mature and develop, and we publish those, then hopefully we'll be able to add those pieces of evidence and checks. So it's just predictions at the moment.
Okay. So another question, a bit longer one. A previous version of ProtVar API had a CSV streaming option. Is that phased out now? It was helpful because I didn't have to parse the JSON out. I could just keep columns as it was parsed by the server.
So we have make significant changes to the API, especially the download API. So you can still download. This is what the download functionality is about now. So if you have your job ID, you can download the CSV. So now it is a compressed CSV. It's not an uncompressed as it used to be. So basically, yeah, we've just tried to optimize this and change the flow around how user download results from the API. But it is still there, but just the way how you get it has changed slightly.
Okay, thanks. Okay, we have a few more questions, but as we have 5 minutes left, we'll still try to go through them. But if I can ask you, James, to move to the next slide. So I would just like to let all our attendees know about the upcoming webinars that you can already sign up on the website. I'm just putting the link now here on the chat. So on the 8th of June, we will have a webinar on improving the digital accessibility of websites using the EMBL-EBI website as a use case and showing how we managed to transform the website significantly in that aspect. And then on the 21st of June, we will have a webinar on identifying suitable patient-derived cancer models in cancermodels.org. So yeah, and you can also watch the recordings from previous webinars in the link that appears also below, where we will also put the recording of this webinar. Okay, moving to the last few questions that we have. Assuming the fold X is folding stability predictions, will you provide in future binding stability predictions as well?
Yeah, it'd be a nice thing to do. It's another thing that we're actively doing with our collaborators. Yeah, because we have the interfaces and we fold them together. So we use the collaborators use the AlphaFold algorithm and to fold the two chains in each other's presence. So we could add to that potentially the missense variant and see how that affects the stability of the interaction itself. Yeah, that's something that would be nice to do. I'm glad you're interested.
Okay, perfect. What's your recommendation for mapping large VCF files, such as the new release of Nomad coming later this year?
Yeah.
Calling VCF strings 500 at a time, or is there a more robust way?
Yeah, so like I say, we'd like to make these big ones available. Splitting a really huge thing that's maybe 50 million or so up would be sensible, but certainly we wouldn't need to split it into 500 at a time. Perfectly reasonable to submit 50,000, even 100,000 at a time would be fine. I mean, submitting 20, 30, 40 million may well be fine. It might just mean that you're waiting for several hours or a day in the queue to be able to process all of those inputs. But like I say, for the very big ones like that, our idea would be to make them available as an FTP download so that you're able to retrieve them without having to wait all that time.
How do you differentiate? Oh, you might want to read this one. How do you differentiate GRCH37 from GRCH38 when the user provides genomic coordinates? It could mean different genes, proteins in both versions of the genome. I think this refers to one of the first questions that we had.
Yeah, I mean, it's extremely important because if you submit GRCH37 coordinates and you submit them as 38, as the user says, it might point to some sensible place in the genome, but it'll be completely the wrong gene, the wrong protein. So the way we do it is we look at the reference nucleotide that the user's provided, and then we compare it with what we expect to be there at that position in the assembly in 37 and in 38. If it's the wrong assembly, we expect it to match approximately 25% of the time just by chance, for example. Whereas if it's the correct assembly, it will be much closer to 100%. It usually won't be 100% with a large set, but if one of them's 30% and one of them is 95%, we make the assumption that it's that assembly. And then we explain to the user on the next screen that we've matched this number to this build, and we think it's this assembly. That doesn't work, of course, very well if you only enter 1 or 2 or 3 coordinates. We really can't, it's not statistically powerful enough to know which build it is. So entering a single coordinate, we really wouldn't have any confidence knowing which build it was. But as long as it's at least, for example, 10 or more, we should be able to fairly reliably know which assembly it's from.
Okay, perfect. So I think that was the last question we had. There have been also a few comments saying that congratulations, thank you very much, great work, and that it's a great tool. So as the presenter said, everyone is encouraged to really use it and get in contact with James and Pahat about possible questions that you have. So as we are over the hour now, I think we will close it here. But thank you, James and Pahat, and thank you everyone for attending this webinar. And have a nice afternoon.
Thanks everyone for coming.
Thank you. Thank you, everyone. Bye.
Bye-bye.